Good evening and welcome. My name is Adam Wronski, and today I'll be discussing investigating observations of socially polygynous behaviors in the eastern bluebird by a genomic approach. The mating systems of birds have been extensively studied, and several distinct categories have been observed. A 1995 study by Johnson and Burley found over 15 distinct categories of mating behaviors. Overall, polygamy is quite rare. About 95% of bird species are normally monogamous. Before we truly get started, I should explain the difference between the three terms polygyny, polyandry, and polygamy. Polygyny basically means a male mating with multiple other females at the same time, whereas polyandry means a female mating with multiple other males. Polygamy, on the other hand, is a more general term that simply describes an individual mating with multiple other individuals at the same time. Within polygamy, there are actually two different types, social and genetic polygamy. Genetic polygamy is a more broad term, again, just describing any individual mating with multiple other individuals at the same time, and they have no interactions beyond this initial mating interaction. Social polygamy, on the other hand, describes when an individual forms multiple pair bonds at once. So in addition to mating with multiple individuals, they will also participate in activities such as care for young, such as feeding them, or defensive territory. Social polygamy in particular is interesting because of the effort it requires. Normal polygamy makes a lot of sense evolutionarily. Mating with multiple individuals generally means more offspring and also higher genetic diversity within those offspring. Social polygamy achieves the same effect, but it puts a much greater strain on the polygamous individual as there are more mouths to feed and much more territory to defend. It's estimated that only 1 to 2% of bird species practice social polygamy at an appreciable level, so it is quite rare in species in the wild. However, experimentally, there have been times that they've been able to induce polygamy into wild species. Kind of one classic study was carried out in 1984 by Wingfield, in which he induced polygamy in 2 species of sparrow by increasing their testosterone levels. Some slightly more recent studies done on a warbler species and a red-winged blackbird basically found a link between nest site and nest box availability and polygamy. And there's kind of this idea of the polygamy threshold model, basically saying that once a certain factor gets to a high enough level, whether it's food availability, nest box availability, whatever it may be, once that threshold is reached, there's a far higher chance that polygamy will occur. The eastern bluebird itself is one of the best studied of all the birds in North America and is a favorite of scientists and non-scientists alike. There's some threat to its habitat as humans have expanded out into its natural range. However, this has been largely mitigated by the creation of bluebird trails across the country. As I previously mentioned, social polygamy occurs in other species, but not very frequently. Usually around 1 to 2% of bird species are believed to be polygamous, and it's often been linked to mate or nest site availability in the cases that it has occurred. Extra pair mating was first observed in the eastern bluebird around 35 years ago, and it's now known that in some populations, up to 15% of females care for offspring that are not their own. At the time of this discovery in 1984, this was quite shocking, but like I said, it's quickly been realized that extra pair paternity is quite common in birds. Recent surveys put it at anywhere between 0 and 76%, depending on the species that you're looking at, with an average of about 16% extra pair paternity across all bird species. The other shocking thing about these initial observations in the eastern bluebird was that it was among the first of these extra pair interactions in birds to actually be confirmed using genetic data. So on Rippon's campus, we have a bluebird trail with about 75 nest boxes spread over varied habitat, including a restored prairie, a golf course, and just backyards of residences. The birds in each nest are banded and tracked, including whether they return each year and their reproduction, so we know, who are the offspring of which individuals and things of that nature. In the summer of 2017, observers on the Rippon Bluebird Trail saw the individual to the right here, a male, going back and forth between 2 nest boxes, box 10 right here and box 14, which are located near the Rodman and Wilmore centers. He was seen participating in typical pair bond activities at both nests, such as the feeding of young and defensive territory, and both of these nests went on to produce offspring. It's interesting to note that in addition to the polygyny that seems to be occurring, this male that was going back and forth between these nests is also the mother of the or is the father of the mother found in nest 14. This seems to indicate that in addition to the polygyny that's occurring, there's also incest occurring, and we'll discuss that a little bit later. This observation was significant because although extra-pair copulation is relatively common in the eastern bluebird, social polygyny of this nature has only been seen a couple times and has never been confirmed genetically. So, following these observations, we wanted to get a look at the genetic data. So we started with the hypothesis that genetic information would be indicative of the occurrence of social polygyny in this population of eastern bluebirds. To begin this research and to kind of look into this topic further and attempt to find some data, we needed DNA sequences. To accomplish this, we used Minion, which is a nanopore sequencing technology. Nanopore sequencing has been around since about 1980, but only recently has it become commercially available in this smaller form of Minion. It operates by pulling single strands of DNA through microscopic pores. Each Minion has several thousand of these pores. The strands are pulled through one nucleotide base at a time, and each base that's pulled through gives off a unique charge signal. Thanks to cloud computing power, these charge signals are turned into a DNA sequence, and it's actually done quite rapidly. As I mentioned, this is a relatively recent technology. Minion was actually only released in 2012, but it's already proved very useful. During the Ebola outbreak in 2014, it was able to give the entire genome of the virus in under 24 hours, and it's even been used in space. Additionally, it's worth mentioning that this device only costs $1,000, which compared to other sequencing methods where you need, an entire lab's worth of equipment, is very affordable. It's also just a small box about this big, and it only weighs a couple of pounds, so very easily transportable as well. So to begin to obtain some samples that we could process through our Minion, we actually got blood from the offspring of both of the nests and interests, as well as the mother of box 14. We would have liked to have got samples from the other two parents, however, we were unable to catch them. Once these blood samples are obtained, they were frozen. When it eventually came time to look at them, they were thawed out, and then we extracted the DNA from those blood samples. That DNA was then processed and cleaned as described by the Minion protocol. It was then loaded into the Minion, where it was read and base called using Epitome software. We then aligned those sequences that we got from that with reference microsatellites from GenBank, and then analyzed base pair differences in microsatellites between individuals. This sounds kind of simple from these directions, but this is really the 10,000-foot overview of the whole process. Everything up to and including base calling with Epitome actually worked very well. It was very quick and we had very few problems. However, after that, there are some complications, which we'll get to in a minute here. Microsatellites are areas of DNA where the same or similar sequence is repeated several times. There's usually between 5 and 50 repeats just stuck end to end in the genome. And microsatellites were used because they're highly polymorphic, which means that they're good for determining relatedness in a group of very closely related individuals, such as we had in this population. So next we get to the point where things get kind of complicated. On the left here, we have the list of some of the reference microsatellites available from GenBank. All in all, there are about 200 of these total that we analyzed. And then on the right, we have the raw data from Epitome base calling. To the far right here, you'll see the start of some of the sequences for these respective individuals. And on the far left, you'll see these barcodes. Barcodes were basically short sequences that we added to each individual's DNA so we could keep track of them while running them through the sequencing and know basically what sequences belong to which offspring. The main challenge once we had all this data was to get it all aligned in some kind of reasonable way. There's several different options out there to do that with various softwares, and they all give different alignments, but the general consensus seems to be that Epitome is the best option. Following these alignments, we got several alignments that looked quite a lot like this one on the screen. In total, we had 10 microsatellites that aligned with any of the individuals. However, there were only 2 microsatellites that aligned with offspring in both nests, as well as the mother of nest 14, which again was the only parent that we had available. The main problem was that we couldn't align each of the alignments together without any coding knowledge. So we basically wanted to stack them on top of each other so that we could compare, but we had basically no way of being able to do this. Additionally, most of the microsatellites were far longer than the sequences they aligned with. You can see this over here on the top, the colored pieces, that is our reference microsatellite, and the lower part with less color in it is the actual sequences we got from the minion. And at that beginning right here in the far end, you can see the microsatellite stretches much further than the actual sequence that we got. This was really messing up our statistical analysis because it was basically counting these as being unsimilar, even though we had no data for our actual sequence that we were comparing there. Finally, basically by typing all of these sequences out by hand, we were able to actually get the multiple alignments where you could see all the individuals compared to each other. However, we still had the problem of the ends not really lining up, as you can see right here. To fix this, it basically involved just more editing of the genomes by hand. So we basically cut it down to the point where each of the sequences had a nucleotide and called that our start, and then went to the end, and wherever the shortest one cut off, that's where we cut all of the other ones off, just to make sure that we are only comparing places where we had actual data. From this, we were able to create similarity matrices for our two different microsatellites that match to individuals in each nest. This one shown right here had the most individuals that align to it, and the main column of interest is the second one to the right here, showing the box 14 mother compared to all these other offspring. You'll notice that she was 59% and 63% similar to two of the offspring from box 14, and 55 and 68% similar to the two offspring from box 10. Looking at other microsatellite here, you'll see again in the second column that the box 14 mother was 96% similar to the offspring from box 14 and 87% similar to the offspring from box 10. Finally, we combined the two sequences end to end so that we were comparing everything all at once, and as expected, this basically gave us an average of the two similarity results from those two other matrices that we already looked at. The main problem with this data, as you may have noticed, is that there's really no clear trend and that there's very little difference in similarity between the mother and offspring from each nest. In fact, even for the first microsatellite we looked at, the mother sometimes was more similar to offspring from box 10 than she was to her own offspring from box 14. In general, we would expect the mother to be at least 50% similar to her own offspring, but like I said, she was also extremely similar to offspring from box 10. As I explained earlier, this data was all kind of complicated by the fact that the mother of box 14 is also the offspring of the male that reproduced with her and the female in box 10, so there's that factor of incest as well as polygyny kind of interfering with our data and calculations. In an ideal world, we would determine the parentage using an exclusionary power calculation, which relies on a series of other factors, including the rate at which nucleotides change in the genome. However, you can't do this for the eastern bluebird at this time because there is no reference genome available yet. So the main conclusion from this research, we really wanted to see if this type of study assessing parentage could work with simplified methods, especially evaluating the use of minion. It really did work well, and minion was definitely promising as a tool. Future research into this subject has the potential to be extremely impactful on human understanding of bluebird reproduction, as you would have the opportunity to confirm social polygyny with genetic methods for the first time ever in the wild. Future studies may want to try to induce polygyny by some of the previously described methods, such as altering the testosterone levels of the birds or changing their habitat. Also, these studies could benefit from using fresh blood samples, as we believe some of our samples lost their quality of their DNA because they had been frozen for so long. Also, it may be beneficial to compare your samples in your study to an entirely unrelated female. As I mentioned, we had several complications because we were comparing to parents that were taking part in incest as well as potentially polygyny. Finally, the literature says that most studies of this nature look at several hundred microsatellite sites at a minimum in order to reach any kind of conclusions. And we think that with higher quality samples and higher quality DNA, you would be able to have at least a few hundred microsatellites that you could look at. Finally, a reference genome is on the way thanks to UW Oshkosh and Dr. Lisa Dorn, so it may be possible in the future to do some type of exclusionary power calculation. As I mentioned, the Minion technology actually worked great. It's an excellent, cheap alternative to other sequencing methods, and it really levels the playing field in terms of citizen science and can be extremely useful as we've seen in crisis scenarios. Finally, I think the main lesson I learned from this is that genomics as a field is relatively new and the software is very much still in development for the most part. As I mentioned, there were countless hours that I wasted basically just trying to line up sequences by hand, when if I had known some minimal coding, I probably would have been able to do it in 5 minutes. And a large part of this is that there aren't any well-developed programs for genomics. There's nothing where it's really easy to jump in the deep end and understand right away. So the development of those kind of programs that allow people to access it both at a introductory and a higher level would be extremely beneficial in the future. Finally, I need to thank Dr. Khan for providing the inspiration and data for this entire project, as well as the encouragement and support to actually follow through on it and get it done. Dr. Sisson for providing her expertise on the complex world of genomics, Dr. Bob Wallace for sparking my passion for research and biology, the entire biology department for teaching and mentoring me so much over the past 4 years, the Bluebird Trail monitors for making the study possible through their hard work and data collection, and Dr. Lisa Dorn and Travis Smith at UW-Oshkosh for their guidance and expertise. I look forward to receiving your questions, and thank you.